All right, hello all. Welcome back for another week of the zoology lectures. And for this week, we'll be going over homeostasis and osmoregulation and the different roles it plays in living organisms. So to begin with, we'll start with homeostasis. And so homeostasis is really just maintaining a constant internal environment. So this process involves controlling variables such as temperature, the amount of salt in your body, even the amount of water or glucose. So the internal environment of your body changes a lot. So stomach is constantly absorbing new food. That changes pH, so you have to excrete more acids or enzymes. The pH of your blood changes very slightly, but it does change from time to time based on how much exercise you're getting. If you're running, the pH of your blood increases because your body's absorbing oxygen and creating CO2 much faster. So it's important to maintain an constant internal environment, or at least as close to it as constant as it can be, because if you have this wildly different external environment, it can lead to denaturing of proteins, which is extremely bad for your cells. So one of the ways that you maintain that constant internal environment are through alternate forms of enzymes called isozymes. And so enzymes function at a very specific and rather narrow set of conditions. When you have a body that exceeds or goes under those conditions, your body begins to produce what are called isozymes, and those are the alternate forms. And so, to give an example, if you're running again, your body is using more oxygen than is readily available, and so you start to undergo anaerobic respiration. And so at that time, lactate dehydrogenase kicks in, so that's an isozyme, and that starts producing glucose much more readily than, I forget what the regular enzyme it replaces is, but lactase dehydrogenase is an enzyme in this scenario, or an isozyme in this scenario. And so energetically, lactate dehydrogenase and other isozymes are extremely expensive to produce. So it's kind of a cost-benefit comparison. Enzymes are readily available, but they only function under a very narrow set of conditions, whereas isozymes are extremely expensive to form, but they can function outside of those conditions, such as higher temperature, lower pH, so much more acidic, or even higher or lower salinity than the organism's used to. So if you live in an area, or if you're an organism like a fish that lives in an area where the environment is constantly changing, and your body has to keep matching those conditions, you have to produce more isozymes, and so that is extremely expensive on an organism. It costs a lot in terms of the energetic requirements that is necessary. So you have to eat much more and stuff like that. So there's a couple basic terms that we'll have to go over first before I continue. And the first one is the solute. And so if we take a cup of water and we have some salt with us, and we mix the two, salt becomes the solute. So we're dissolving salt into water, and so water is the solvent. Both of those two chemicals together, salt and water, that becomes a solution. So once again, salt's the solute, water's the solvent, and then both of those together is a solution. Osmosis is when we have water that's moving across a permeable membrane, such as the lipid bilayer in cells. And so this is driven by a difference between solutes, not solvents. So diffusion is driven by the concentration of solutes. So once again, osmosis is the difference in solvents. Diffusion is the difference in solutes. So in diffusion, let me get the pen up here, you have a higher concentration of this solute right here, and another concentration of solute right here. So you have 1 and 2. Those are at 2 non-equal concentrations. So you have a concentration of solvent 1 here, and you have a concentration of solvent 1 or 2 over there. If you think of that in like some sort of anthropomorphic sense, they both want to intermingle and be equal with each other. So they'll diffuse across this permeable membrane. So we have solvent 1 here moving across into this second area, and they have solvent 2 moving into this area here. Once they've each have reached an equal concentration on either side, so you have both 1 and 2 on both sides, that's what we consider an equilibrium. So this word right here, that's basically what I just said is exactly what it means. There's an equal concentration on both sides of a gradient. So that's diffusion. So diffusion is solutes, not solvent. When we move on to osmosis, we're talking about solvents. So in this case, we're talking about water, but it could also be whatever chemical structure you're thinking of that is a solvent. So I believe ammonia could be a solvent. Yeah, mine's blanking on other solvents at the time, but water is the most important one for life on Earth. So in this case, we have 2 layers or 2 sides of a semi-permeable membrane. So we have this lipid bilayer right here, where water can transport across freely. So if we start with an area with lower concentration of solutes, which is right here, and then there's a higher concentration on the second side there, water will actually flow to the area with the highest concentration of solvents. So if you think about in terms of cells, if your cells have a high concentration of salt, water will flow into it. If your cells have a low concentration of salt, but the water outside of it has a high concentration of salt, the water inside your cells will flow outside of your cell. So you can kind of think of it like a pile of salt. If you leave salt outside of its container, so if you have those big jars of iodized salt, they start to clump up. That's because water is coming outside of the atmosphere and going to that area of high solvent. So that's not exactly an example of solutes, but or a closed environment like we're talking about here, but you get the picture of how salt being the solvent or solute is attracting the solvent water. So you might have done this in high school, but you can actually test this using a test tube with 2 open sides and then a selectively permeable membrane. So this right here could be a balloon. Balloons work well. Latex works well as well. So if we take distilled water, and so distilled water is pure water. There's no other solutes in it. It's pure H2O. And if we take a salt solution, so water mixed with salt, and we set this structure into that jar of distilled water, that distilled water level inside the jar is going to drop, and the level of water inside that test tube is going to increase. And this is because in that test tube, if I can draw it here, you have a high concentration. If you imagine all those lines are salt, and this distilled water, you have a very low concentration of solutes. So water is going to flow from low solute concentrations to high solute concentrations, so water is flowing up this test tube, causing that rise in osmotic pressure. So this happens in your cells. I mentioned this earlier. If you have a high solute concentration in your cells, water will flow out of your cells into other, I believe that's, yeah. So if you have a high solute concentration in your cells, so imagine if you have a high salt concentration in one of your skin cells, and you stick your finger into, let's say, salt water, your skin cells start to shrivel. If you stick your finger into fresh water, your skin starts to swell. That's why you prune up in the shower. So once again, you have a low concentration of solutes on the outside, and then you have a higher concentration of salts on the inside. So when you're in fresh water, water will flow into the cell, and the opposite is true. So if you have a higher concentration of solutes on the outside, water will flow out of your cells. So there's special terms for this, so you can ignore the plants. This isn't botany, this is zoology. So when water is flowing into a cell, so there's a lower concentration of solutes outside the cell than there is inside the cell. Water flows in, and that animal cell will burst, and so we consider this a lysed cell. There. If our cells at equilibrium, so if there's both equal gradients or concentrations of solutes on either side of that membrane, we consider that a normal cell. And so if we take this looks like a normal blood cell. So when there's a equal concentration of solutes on either side of a cell, so inside and outside, we consider that an isotonic solution. I forgot to mention it on the last one, but if there's a higher concentration of solutes inside a cell than there is outside, we consider that a hypotonic solution. And if there's a higher concentration of salts or solutes outside the cell than there is inside the cell, we consider that a hypertonic solution. And so when that cell undergoes, there's dropped in the solution, it becomes shriveled because all the water is flowing out of it in order to maintain, or through osmosis. So there's two very basic definitions for the types of organisms that live in these very variable environments. If we have an organism such as this spider crab right here, found in the deep sea, it lives in very stable environments. There's really not much change. Salinity is not changing as much. It's at very deep depths. Temperature doesn't change as much. It stays fairly cold. I believe right around 35 degrees Fahrenheit, roughly 1 degree Celsius. And then all other chemical compositions in the water also stay fairly constant as well. So we consider this a stable environment. There's really no change going on. This organism doesn't have to expend a lot of energy creating those isozymes, those alternate forms of enzymes, because it doesn't have to survive those extremes, and it's really not subjected to those extremes. So organisms in this environment we would consider stenohaline. They can tolerate a very narrow range of salinity. They're not used to fresh water, they're not used to brackish water, they're used to this very narrow range of salt water. So if you took that spider crab and you placed it in a tank of fresh water, all of its cells would lyse, so it has a higher concentration of salt or solutes inside of its cells. Because of that, when you drop it in the fresh water, all that fresh water is going to flow into its cells, and so its cells burst, and then it would suffer a terrible death. The opposite is true for organisms that live in extremely unstable environments. So if they live in tide pools or estuaries where the tide will raise, those tide pools fill up with water, and then as the tide drops, those tide pools are left out in the sun. So as those tide pools sit out in the sun for the majority of the day, the salinity actually increases in those pools because the fresh water is evaporating, leaving the salt behind. So you have extremely high salinity in these tide pools. And so the same thing also happens in estuaries. You get this influx of fresh water, and then as the tide flows out, you have higher salinities that's left behind. So the organisms in these sorts of environments are considered osmoregulators. They're actively controlling the conditions inside their body. So when we're talking about salinity, these organisms are considered euryhaline. So if you take a shore crab or a blue crab and you put it in a tank of fresh water, you put it in a tank of brackish water, or even a tank of salt water, it will survive each of those immersions because it is actively controlling its internal environment. So its cells are actively matching the external environment. So if we take these two organisms, the shore crab from the tidal areas, and you take the spider crab from the deep sea, and then we plot the salt concentration inside their body and the salt concentration, well, I'm covering x-axis, but the salt concentration outside their body. So let me just write out. Yeah. So you can see that spider crab, the one that we consider stenohaline, survives a very narrow range of salinities. It only tolerates from roughly 400 to about 450 or 650, whatever units of salinity this is. So normal seawater is roughly around 30 parts per thousand. So you have 30 milliliters of salt to 970 milliliters of water in a liter of ocean water. So I think these are in millimoles. And I'm not sure what the conversion is for that to parts per thousand or milligrams per liter. But you can see the stenohaline, when it's subjected to changes in seawater or salinity, it doesn't survive outside of this range. So these crosses indicate that it died at 400 and then 650 millimoles of salinity. If we consider our shore crab, which is found in those tidal environments, and it's euryhaline, it survives at much more or much lower amount of salinity, so roughly about 180, and I can't see the x-axis, but around 700 millimoles of salinity. So you can see up to a certain point here, it's actively controlling the amount of salt inside of its body. So it has these adaptations where it can control the amount of salt inside of its cells. So as salt water is increasing, the amount of salt inside of its body doesn't really match that increase until we get to this point here at around 400 or 500 millimoles of salinity. So from here to here, ignore that line. From here to here, it's actively controlling the amount of salt in its body, and then from that concentration of seawater to here, it's matching the salinity. So technically, if we want to consider it both an osmoregulator and an osmoconformer, so osmoregulator. So it's osmoregulating from 180 parts per thousand or millimoles to around 500 millimoles, and then it's osmoconforming from 500 millimoles all the way up to its death at roughly 700 millimoles. The spider crab was an osmoconformer the entire time of its life, so its internal concentration of salts matched the external concentration of salts, so that was a true osmoconformer. So the osmotic balance, I completely ignored that, but it's at the top of this slide. So an osmotic balance is just when there's it's when the amount of solutes on either side of the body are equalized and creating an equilibrium. So here, if you take that spider crab, you have a equilibrium on either side. If we take where it died, at the lower end of its freshwater or fresher water, the lower end of its tolerance to salt, at 400 millimoles and 400 millimoles, we get this basically a line with the slope of 1, so perfectly diagonal. And same thing if we take where it died, it goes all the way from about 650 to 650. So that's a true osmoconformer, and on either side of its body, so internal and external, it's maintaining that equilibrium on water concentration. So moving on from that, we'll talk about the excretory system. I'll go through each of these structures. So I'll go through the Protonephridia in the organisms that we just saw on Wednesday and Thursday of last week. So in the planaria, there's the Protonephridia, and then each of these groups I'll try and cover a little bit in detail when we get to them, but for the rest of this presentation, I'll try and go over them in a lot more detail than I will later on in the semester. So the first one is the Protonephridia, and so these are found in Platyhelminthes. There's also a very similar structure found in chordates, and we'll get to that in 6 weeks. And so those are found in lancelets. And so Protonephridia is an extremely basic excretory system, and so it just takes these flagellated cells called flame cells, and it just moves waste products from the body to an exit pore called a nephridia pore. So those flame cells are called flame cells because those the flagellated portion of it looks like a flame while they move water past it. So one of the hypotheses on how Protonephridia first arose is that it was a way to match hypotonic environments. So remember, hypotonic means there's a higher concentration of solutes inside the cells than there is outside. Water would be flowing inside into this organism. So this means that the first Protonephridia likely arose as a way to keep the amount of water inside the body low, and then remove any of that excess water that keeps entering that body. So the second group or type of excretory system is called Metanephridia, and so this is really just one step above that found in the Platyhelminthes, the Protonephridia. So this is found in annelids, so segmented worms, and then mollusks. I think I spelled that wrong in this PowerPoint. Yeah, well, don't ignore my spelling. So this is when there's some sort of direct opening from the body cavity to an external environment. So we have here what's called a nephrostome. So this is this funnel that has cilia, and it's connected to each of those body cavities. So throughout this organism, there's this long tube that's open. That's the body cavity. Waste enters these nephrostomes and is pulled along by that cilia. So that cilia is constantly undulating, creating some sort of water current. All waste products are transported into this nephrostome. And so that nephrostome and all those waste products flow through, passing through this capillary bed where even more waste products enter, and then from there it exits through a nephrostome. So still very simple. Each of these groups, so there's multiple structures like these throughout the body of an annelid. Each of those groups is called nephridia, and so you'll have one basically per segment along the entire digestive tract of this this earthworm. For aquatic arthropods, such as crayfish or lobsters, they have what are called antenoglands. And so ammonia is passed into a bladder, such as this one. And then it passes through a series of tubules, enters a labyrinth, and then it exits out of the second antennae, the base of the second antennae here. So ammonia, which is a fairly toxic substance, which we'll talk about in a couple minutes, will be passed through into the bladder through the circulatory system and a little connection to the digestive tract. It passes through these tubules where all fresh water is removed. So it's keeping fresh water inside its body and then passing ammonia outside of its body out of that end sac, which is right here at the base of that second antenna. That's why it's called an antennal gland. It used to be called a green gland, but they changed the name to antennal gland. And it's named that because the exit is right by the base of its second antennae. So you have the first antennae here, and you have the second antennae here. So, in insects and other terrestrial arthropods, such as insects, myriapoda, like the centipedes and millipedes, we have what are called Malpighian tubules. And so these Malpighian tubules connect directly into the digestive tract. So if you look at this diagram here, each one of these tubes here, that's a Malpighian tubule. And so salt, water, nitrogenous wastes such as urea, ammonia, uric acid are transported from the circulatory system into the digestive tract. And so all that waste exits the body as feces. Kind of a neat factoid is that inside the rectum, any other particles that are being passed that are still necessary for this organism, such as water, some ions and electrolytes, and other organic molecules, can get reabsorbed back into the circulatory system. And so if this insect lives in a very dry environment, such as a desert, it will continue to recirculate water throughout its body until it's no longer usable. So water will it'll drink water, it'll go through the intestinal system and get passed. But before it does, it passes through the rectum, and if that water is still usable in a usable form, it gets reabsorbed back into the bloodstream, and it starts over again. So if there's excess water, it just passes out that way. In chordates, we have kidneys. Well, most chordates have kidneys. Most of the fish, so Actinopterygii, the ray-finned fishes, don't have kidneys. They live in an aquatic environment, so they can just pass their nitrogenous waste out of their body without really worrying about without it building up inside their body. However, most terrestrial organisms do have to worry about this. Ammonia is a very toxic nitrogenous waste, so we convert it to urea, and then we still have to store it inside of our bodies until we can pass it. So inside of our kidneys, we have what are called nephrons, and so these act as a sort of blood filter. And so blood enters through a renal artery, which is shown. Well, we have a renal vein right here, which is this blue structure. The renal artery is just below it in this pig dissection. The kidney is this structure right here. It's a big circular structure. Looks kind of like a lima bean. And so once again, that kidney's acting as a filter for the blood. It's storing all that waste until it can get passed to the bladder. So put a K for kidney. And so the bladder in this pig is this little balloon-like structure right here. Put a B for bladder. So that's a terrible B, but anyway, before urine is created by the kidneys and passed to the bladder, it's that kidneys acting as a filter, and so blood enters through that renal artery, and then all separate fluid, so all that waste fluid is separated from the blood through what's called a glomerulus. And so inside each of these kidneys are several thousand what are called nephrons. And inside those nephrons are different structures. I forget the 3 major types, but this isn't really a anatomy class, so you don't really need to know which structures are inside each nephron or inside the kidney. Once waste is produced through the form of urine, it passes through a ureter, which is this tube right here. And so that goes to the bladder, and then the bladder stores it until the body can relieve itself. So yeah, that's the kidney. I mentioned that chondrichthyan fishes don't have kidney, or they do have kidneys, whereas Actinopterygiae, the ray fin fishes, don't. Chondrichthyan fishes, so sharks, rays, and skates, they all produce urea, and so they have to have a complex excretory system, such as kidneys, to excrete that waste. Other organisms like Actinopterygiae, Petromyzontidae, they just excrete ammonia, so they don't need to have these kidneys or these complex excretory systems. So I keep mentioning ammonia, urea, and uric acid, but we really haven't talked about what they are. So ammonia is this really basic structure here, made of 1 nitrogen atom and 3 hydrogen atoms. And so this is one of the base products of metabolism, also one of the base products of creating nucleic acids. It's a very toxic substance, and so you can't really store this in your body. So we have a bladder. If we stored ammonia in our bladder, we would die from basically being poisoned by our own waste products. So we convert it to urea, which is the structure here. Takes a lot of energy, but it's much more safer for our bodies. So we can store urea for short periods of time. Uric acid is formed, and you can see it's a very complex structure. It has these carbon rings and double bonds. So this is extremely energetically expensive to produce, but it's not toxic, and it saves a lot of water. So a little bit more detail. So ammonia, like I mentioned, is produced through metabolism. So when you burn off ATP, you're using energy, you're building proteins, you're making nucleic acid, you're forming ammonia as a waste product. This is, as I mentioned, is the most toxic nitrogenous form. It's extremely basic, and it does a lot of damage to your cells. When you live in an aquatic environment like a fish, you don't have to worry about storing ammonia. You don't have to worry about converting it to another chemical structure. You can just simply diffuse it out into your environment. So fish will do this through their gills. Other aquatic organisms, such as the Daphnia that we were looking at on Wednesday or Thursday will just pass ammonia out through through their Antenoglands. And so this is one of the reasons why if you have a tank of freshwater fish, you have to change the water. You can't just let them be, because as those fish are living inside that freshwater tank, they're continuously producing ammonia and just simply diffusing it out of their bodies into that water. Over time, the amounts of ammonia will build up inside that tank, and it will become poisonous to the fish. So they'll be basically swimming around in their own metabolic waste and poison. Another thing about ammonia is that it loses a lot of water when you're just diffusing it out into the environment. So you also have H2O being lost as well when you're just diffusing ammonia out into the environment. So there's no true or really complex excretory structures that are holding back that water. And so when you're living as a fish in a freshwater environment, you don't really care if you're losing water because you're surrounded by it. There's no real need to have a storage system for water or reabsorb water when you can just float around and move to a new spot and there's fresh water there. Terrestrial organisms have or don't have that ability, so we have to maintain a certain level of water inside our bodies in order to survive. So we can't afford to just excrete ammonia and lose all that water. So we convert it to urea, which is a less toxic form of that nitrogenous waste. It takes a little bit more energy, but it's less toxic, and we don't lose as much water in this process. The most expensive or energetically expensive form is uric acid, but there's almost no water lost in this process. So if you've ever seen a car, or if you've ever owned a snake, that white stuff you see here, that's all uric acid produced by birds or reptiles or whatever. There's almost no water lost here. It's basically the most efficient way to save water for an excretory system. It is the most expensive to make. It requires a lot of energy because it has these complex carbon rings, double bonds, here, and here. So it is complex, but it does save you water. So if you're living in a desert environment and you can't afford to lose any water, it hasn't rained in a week or 3 weeks, and you need to save water, this is the way to go. Some terrestrial mammals that live in deserts, I believe, also create uric acid instead of urea. I think it's like the kangaroo rat and some other rat in the southwest. So it's an extremely expensive process, but once again, saves water. So if you want to look at this chart in a more simple sense, you have increasing energy cost in this direction. You have decreasing toxicity in this direction, and you also have decreasing water loss. So at ammonia, you have the lowest energy cost. It doesn't take a lot just to diffuse ammonia out into the external environment. It does lose a lot of water in the process, and it's highly toxic. When you have urea, it's moderately toxic, but you have less water loss, and it's really efficient in terms of energy cost. Then you have uric acid, and so that's the most expensive to make, but you're saving the most amount of water, and it's pretty much non-toxic. So when we get to lab on Wednesday or Thursday, we'll be testing the theories of osmoregulation and homeostasis on a potato, and you can't see it, but a goldfish, or goldfishes. So when you come to class, work close to a choose. We'll be working with live animals again. Make sure you take the quiz. It's due Wednesday at 12 or noon. Yeah, I'll see you guys then. If you have any questions, make sure you ask them on that discussion board on Blackboard, or just bring it up before we get started in the lab. So.